Good evening, everyone. I welcome you all on this webinar on a very interesting topic on biomarkers in immunotherapy for solid tumors. We are Team MedGenome, the leaders of genetic diagnostics in India. So without wasting any further time, let me introduce our speakers for tonight. Our first speaker is Dr. Nirmal Raut. Dr. Nirmal Raut is a senior consultant medical oncologist at Bhaktivedanta and Jupiter Hospital. He has around 20 years of vast experience and he has completed his MBBS from KEM Hospital, MD and senior residency from Tata Memorial Hospital. After having cleared European certification of medical oncology, he has completed his master's in molecular oncology from Spain, and he has various peer-reviewed publications and international journals. Our second speaker is Dr. G Arun Kumar. So Dr. Arun has around 15 years of experience. He is currently associated with MedGenome Labs as the head of technical support and oncology. He has 15 years of experience in human genomics and has a PhD in human population genetics. He has his technical expertise in genome analysis, NGS data analysis, statistical analysis of genetic data, cytogenetics, FISH, acid design of qPCR and NGS in clinical laboratory. So without wasting any further time, I would like to call Dr. Nirmal Raut now for his talk. So over to Dr. Nirmal Raut now.
Okay. Thank you so much for the kind introduction. I will share my slides. Yeah, the topic for today is biomarkers in immunotherapy. If you go to any medical oncology conference today, the most of the clinical trials are focusing on immunotherapy, and why should it not be? The immune checkpoint inhibitors, they have changed the scenario since they have come into the market. So immune checkpoint inhibitors offer survival benefit compared with historical treatment approaches. As you can see in this metastatic melanoma, before 2011, we were very afraid to treat this particular tumor because the survival was so poor. But this is the data in 2017 where pembrolizumab plus reduced dose ipilimumab is used, and then the survival is increased. But the problem with immune checkpoint inhibitors are they work only in 20 to 30% of the tumors. So only some tumors respond. The majority of the tumors don't respond, and we don't know which are the tumors which respond and which don't respond. And there are potential for serious side effects with these immune checkpoint inhibitors, and there's also potential for hyperprogression. Yeah, and apart from that, there are also financial issues. Sorry. Yeah, so they are not cheap. Around 1 year's therapy costs around 20 to 25 lakhs, and it goes on for 2 years. So we need to have predictive biomarkers which can tell us which tumors are the ones which should receive immunotherapy. Yeah. So when we give immunotherapy to one particular tumor, this is a typical response curve that we see. So the one in the blue is a durable complete response. The one in the green is durable partial response. The one in yellow is pseudo progression, where the tumor initially flares up in size and then undergoes regression. And the fourth one, the red one, is the hyperprogression, where the tumor actually increases its potential to multiply. So we need biomarkers to understand which of the immune checkpoint inhibitors to which of the immune checkpoint inhibitors the tumors will behave in which of the particular way. So these are the biomarkers which are there. So the ones on the tumor cell are PD-L1 expression, TMB, DDR pathways, specific mutated gene pathways like interferon, gamma, KRAS, STK11, and neoantigen load. Yeah. And in the tumor environment, microenvironment, we have PD-L1 expression, tumor infiltrating immune cells, the immune status of the TME, immune cells with the specific genotypes, phenotypes, and a diversity of immune reporters. In the circulating factors. They are peripheral blood cells. They can be ctDNA, other circulating molecular markers. And there are host-related factors such as general characteristics like the gene, age, the gender, age, body fat distribution. And very important is the intestinal microbiota. And also one of the other factors is host germline genetics. So based on these, we will be talking on these biomarkers. But basically the biomarkers that I will concentrate on is PD-L1, TMB, and MSI high. And T-cell infiltrate and NGS I will just mention in a brief. So, but let me tell you that none of these biomarkers are perfect. When we look at one particular biomarker, we cannot say that like PD-L1, if it is positive, it will definitely respond. If it is negative, it will not respond. It is not that way. So all these biomarkers are like these 6 blind men and the elephant. The medical oncologist is trying to look at just one part of the elephant and then trying to figure out what the elephant is like. So the biological rationale for immune checkpoint inhibition is that there are various checkpoints in the immune pathway. And the first one is the B7 and the CTLF4. So when CTLF4 is overexpressed, then it is binding to B7. And then the probability of the cells, that the T cell and the dendritic cell, the dendritic cell express the B7 and that is binding to the CTLF4. So CTLF4 antibodies can be used here, and based on that, the T cell activation can occur. And once the T cell are activated, the T cells move from the lymph node into the tumor microenvironment, where the PD1 on the T cell matches with the PD-L1 or PD-L2, and based on that interaction, further the immune response occurs. So we have basic antibodies to PD-L1, PD-L2, and PD-L1 and PD-1, which are used to inhibit these particular checkpoints in place. So PD-L1 is also called as CD74 or B7H1, which is the most widely investigated, and it is present on cancer cells, dendritic cell, activated B and T lymphocytes, and macrophages. It's basically protecting the tissue from excessive inflammation and autoimmunity. So this was the Keynote 001 study, which showed that basically these two survival curves are less than 1%, that is PD-L1 negative, and 1 to 49%. So 1 to 49% and less than 1%, the curves are overlapping, but the PD-L1 more than 50% has a statistically significant survival curve. So based on this study, then they started a phase 2 study, open label phase 3 study in 305 patients with untreated advanced NSCLC and PD-L1 expression more than 50% of the tumors. So they started as another study called as Keynote 041. And as you can see in this slides, the hazards ratio is 0.50. So that is a statistically significant progression-free survival. So at the 1 year survival was increased from 15% to 48%. And the median progression-free survival was increased from 6 months to 10.3 months. And this is the overall survival. The hazard ratio was 0.6, but the overall survival is increased from 54% to 70%. So based on that, we have PD-L1 inhibitors which are used in non-small cell lung cancer. For more than 50%, we have only the immunotherapy that is used. And this is a Keynote 189, which showed that overall survival with first-line pembrolizumab plus chemotherapy versus placebo plus chemotherapy in stage 4 non-squamous non-small cell lung cancer. As you can see, the hazard ratio here is 0.49, and that is statistically significant. So, and if we look at the survival curves as per the tumor proportion score, the PDL1 less than 1% or 1% to 49% or more than 50%. All of these values are statistically significant. However, less than 1% is the significance is less, but it is still statistically significant, and that's why it's still used. And this was the same in squamous cell carcinoma. This, the hazards ratio is 0.64. And this was in recurrent metastatic non-squamous cell NSCLC. So they used atezolizumab plus chemotherapy plus bevacizumab. And with that, they showed that the 6 months, 12 months, and 18 months progression-free survival was statistically significant. The hazard ratio was 0.59. And this was the subgroup analysis, where in the intent to treat population, it was 0.6, while this was the only trial which used EGFR, ALK, and the other driver mutations. So this, based on this, this is approved in second line in driver mutation cases. And this was the triple negative breast cancers, where Impassion 130, where atezolizumab plus nab-paclitaxel was used in patients with untreated metastatic triple negative breast cancer. As you can see in the intent to population cases, the survival curves are not very different. The hazard ratio is 0.84, but the p-value is 0.08. But in the PDL1 positive population, the overall survival separates out, the curves separate out. So the p-value is 0.62 here. So based on that, FDA approved atezolizumab for PDL1 positive unresectable locally advanced or metastatic triple negative breast cancer. This FDA approval came in March of 2019. So subsequently, Keynote 045 study showed that overall survival with pembrolizumab versus chemotherapy in post-platinum cases. So first line, they used platinum in metastatic urothelial cancer. And in second line, they used pembrolizumab as compared to chemotherapy. And they showed that overall survival, there was a benefit in the overall survival, and hazard ratio was 0.7. And this was in the cisplatin ineligible urothelial cancer. Since urothelial cancers occurs at a median age of around 65, 70, most of these patients are having a high creatinine. So they are ineligible for cisplatin. So these patients were randomized to single arm pembrolizumab and atezolizumab. So IMvigor 210 and Keynote 52. And 12 months overall survival was 48% and 50%, 58%. And this was in intermediate and poor risk renal cell carcinoma. The overall survival with nivolumab plus ipilimumab versus sunitinib. As you can see here, the hazard ratio is 0.63, which is statistically significant. With nivolumab, the 18 months overall survival was increased from 60% to 75%. And based on that, in April of 2018, nivolumab plus ipilimumab as a combination got an approval for treatment of intermediate or poor risk previously treated RCCs. So, and this was the Keynote 426, where pembrolizumab plus axitinib versus sunitinib was used in treatment naive advanced RCC. So the only difference was they took all the cases, they took even the low risk cases, and they showed that pembrolizumab and axitinib was much better. The 18 months survival rate was increased from 72% to 82%. And as you can see in this forest plot, whether the PDL1 is positive or the PDL1 is negative, it doesn't make any difference. So for PDL1, we have more questions than answers. What is the optimum cutoff? Is there optimum cutoff based on tumor type or antibody type, impact of the tumor heterogeneity on predictive ability, how best to report the PDL1. Is PDL1 better on the metastatic tumor or the primary site? Do PDL1 concentrations vary over time, and what factors affect them? So this is the prevalence of PDL1 expression in various tumor types. So as you can see in lung cancers, the PDL1 is quite high. This is the PDL1 on immune cells and PDL1 on tumor cells. So in NSCLC and RCC, the PDL1 is high, while in pancreatic cancer, gastric cancer, it is low. So it is a very heterogeneous expression of PDL1. And this is how you express the PDL1. The CPS, uh, uh, mentions the combined proportion score, which consists of PDL1 staining tumor cells, lymphocytes, macrophages, divided by the viable tumor cells multiplied by 100. While tumor proportion score, if the PDL1 staining tumor cells upon the viable tumor cells into 100, while immune cells, the PDL1 of lymphocytes and macrophages upon the tumor area into 100. So as you can see, various assays use different methods of reporting PDL1. And this is PDL1 heterogeneous in the same sample of the lymph node. From one part, it is showing positive, and from the other part, it is showing negative. So these are the problems with PDL1 expression. Yeah. Yes. So the two platforms on which PDL1 is basically measured is DACO and Ventana. And for pembrolizumab, you use an antibody called 22C3. For nivolumab, you use 22.8. For atezolizumab, you use SP143, and the other one you use SP263, that is durvalumab. So for each of the agents, there are different platforms and antibodies that are available. Yeah, and there was this blueprint study which tried to correlate between the all the four platforms, and they found that 22C3, 22.8, or SP263, they all are having the same measurement. Only SP142 is an outlier. So SP142 stains fewer tumor cells than the others. And this is PDL1 based on this is PDL1 based on the previous and the later on when the tumor relapses from phase 2 Atlantic study with durvalumab in previously treated NSCLC. So the concordance rate is 74%, and there was increase in the PDL1 19.5%, and there was decrease in the PDL1 6.5%. Whether they received prior TKI or they received prior CT or prior RT didn't make much of a difference. Coming to the next is the mismatch repair, and these are the DNA repair mechanisms. So the base mismatch is insertions and deletions. They are repaired by the mismatch repair genes, and so mismatch repair is basically short stretches of DNA, 1 to 6 nucleotides, randomly repeated throughout the genome, both in the gene and the intergene area. They are present in the introns, promoter region, untranslated coding regions, coding exons, and MSI occurs when the genome gains or losses are more than or equal to 1 repeats. Yeah, and how do we measure the MSI or the dMMR? Is based on IHC. We look at this the 4 markers: MLH1, MSH2, MSH6, and TMS2. We can look at it from at the IHC level, at the protein level. We can also look at NGS, which detects the mutations in these particular genes. And this mismatch repair genes, when they undergo mutation, they lead to error during DNA replication, which leads to microsatellite instability. So microsatellite instability can be measured by PCR or NGS. These are the markers, the Bethesda markers for microsatellite stability. In 1996, there was a conference in which this mononucleotide markers BAT25 and BAT26 and D2S123 and these three are the dinucleotide markers. So yeah. So based on that, they came with this that you should use these 5 markers. But then later on in 2007, they underwent a revision and they found that this mononucleotide markers are more sensitive than dinucleotide. And based on that, BAT25, BAT26, NR21, NR24, and Mono27, these are the 5 markers which are approved. And if one of these markers is missing, then it is a low MSI. And if more than two or more than 2 loci are present, then it is a high MSI. So this is the TCG analysis of 39 cancer types involving 11,139 tumors. So the top 15 tumors which showed MSI or dMMR were uterine endometrial cancer, colon adenocarcinoma, stomach adenocarcinoma, rectal adenocarcinoma, and so on and so forth. And MSI microsatellite instability have greater T cell infiltrate and checkpoint expression. So yeah, so here you can see there are more new antigens associated with higher TIL, and they also have higher level of checkpoint expression in MSI high. Yeah, so based on the Keynote 177 study, which was based on MSI high colorectal tumors, these were the curves which were seen, the overall survival curves, the progression-free survival curves. So in the pembrolizumab arm, there were 54% events, and chemotherapy arm, there was 73% events. And the progression-free survival was improved from 8.2% to 16.5%. So based on that, based on this, the MSI got FDA approval in tumor agnostic markers. So it is based on all the tumors. When you have MSI high, you can use immunotherapy. But in this Keynote 177, this initial part of the graph, there was a crossover. So you can see initially the survival curves drop down, and then significantly it improves over the time. So they started with Checkmate 142, which was a phase 2 randomized trial, to bring that curve a little above. So they used a combination of nivolumab plus ipilimumab, that is CTLA4, in dMMR or MSI high metastatic CRC. And when you use nivolumab plus ipilimumab in these patients, then you can see that the overall response curves is 55%, which includes 3.4% of complete responses and 15.3% of partial responses, as compared to only with nivolumab 31% overall response rates. And the change in the tumor burden with nivolumab plus ipilimumab is dramatic. 78% of the patient had reduction in the tumor burden from baseline with combination therapy. So these are the progression-free survival, and these are the overall survival curves for Checkmate 142. So 9-month progression-free survival is 76%, 12-month overall progression-free survival was 71%, and there was 50% events in the ones which removed nivolumab, and 25% events in the patients received nivolumab plus ipilimumab. And these are the curves these are the overall survival curves. 9-month overall survival rate is 87%. 12-month overall survival rate is 85%. And this is the study of pembrolizumab in non-colorectal cancer patients who have deficient MMR. So you can see that non-colorectal cancer patients, you have 46% of response rates, of which endometrial, biliary, gastric, pancreatic, small intestine, these are the most common ones. So deficient MMR results in MSI high. MSI high or dMMR leads to higher mutation rates, and that leads to higher number of new antigens. Deficient MMR present in many metastatic and solid tumors, so universal testing is warranted. Immune checkpoint inhibitors is associated with clear benefit in dMMR patients, including patients with long-term disease control and pathological complete response. So now coming to tumor mutational burden. Tumor mutational burden is an algorithmic measurement of a subset of synonymous and non-synonymous substitution and insertion deletion mutations in a tumor's genome. How is it calculated? TMB denotes the number of acquired somatic mutations in the coding region of the cancer cell genome, that is all exons of the approximately 22,000 human genes. The total coding region covered is around 1.5 megabase. So if we put the tumors with high tumor mutational burden at one end and the tumor with the least tumor mutational burden at the other, so then there is a curve that is seen like this. So melanoma, lung cancers, lung squamous carcinomas, lung adenocarcinoma, and bladder cancer, these are the ones which have high tumor mutational burden. So tumor mutational burden is not a is a continuous variable, but it is not limited to a categorical cutoff. So single cutoff for continuous variable biomarkers presume the clinical response is evenly distributed within high and low cohorts, and continuous variables distribute the clinical effect across a wide range of biomarker values. For example, 11 and 30 mutations per megabase are both considered tumor mutational burden high, but may have drastically different chances of clinical response rates. So tumor mutational burden is an independent predictive biomarker. So this is tumor mutational burden, the overall response rate to immunotherapy based on tumor mutational burden. This is the particular the graph, and this is excluding MSI high. So there, this is for non-TMB high. So that is for MSI high. And this is overall response rate to immunotherapy based on tumor mutational burden and TMB. So this is TMB high and PDL1 positive. This is non-TMB high, PDL1 positive. This is TMB high and PDL1 negative. And this is non-TMB and PDL1 negative. So initially whole exome sequencing was used, but it was costly, time consuming, and not practical for clinical decision making. So a targeted gene panel using sufficiently large NGS panel is currently utilized to calculate tumor mutational burden. So so this is Keynote 158 trial, which demonstrated TMB high tumors had a better response to pembrolizumab. As you can see here, the partial response rates are around 25%, while compared to TMB low, which is only 5%. And these are the various different tumors. They took around 10 tumors, and then they found out their tumor mutational burden. And the ones which are, so this is the tumor mutational burden high in all the cancers, but in cancers, in individual cancers, the tumor mutational burdens can be different. So anal, biliary, cervical, endometrial. So in endometrial cancers, you have this high tumor mutational burden, neuroendocrine, salivary, small cell, thyroid, and valvular. So this was the tumor 158 trial, which is tumor mutational burden is associated with responses to immune checkpoint inhibitors. At 1 year, the survival was increased from 13% to 26%. And at 2 years, it was increased from 7% to 22%. So based on this data, tumor mutational burden has now gotten FDA approved biomarker for immune checkpoint inhibitors. So it's a tumor agnostic marker. So any tumor having a tumor mutational burden more than 10 by Foundation Medicine has been shown to respond to immune checkpoint inhibitors. So tumor mutational burden has been based on Foundation 1 CDX and MSKCC impact. These are the two approved NGS tests which are used for tumor mutational burden. And so the TMB harmonization working group, it aims to solve the challenge of the TMB standardization. So various partners such as government, diagnostic, academia, operational, and industry. So these have come together and they are forming a group called as the Friends of Cancer TMB Research. And they are doing this workflow. They are doing this analytical validation and clinical validation. And one of the papers has recently been published. So we wait for more papers from them, the group. And this is the blood-based TMB, which is associated with enriched response rate. This data was presented by Kim et al. At ESMO 2018 in first-line lung cancers. So BFIRST is the first prospective study that showed blood TMB. So liquid biopsy, based on liquid biopsy, they took the tumor mutational burden and it predicts response rate based for atezolizumab monotherapy. But here the cutoff was more. In blood-based TMB, the cutoff was 16 mutations per megabase. Yeah. With that, I end my presentation.
Thank you, Dr. Nirmal. That was indeed a very interesting talk, and I'm sure our viewers have learned a lot about the biomarkers like PDL1 and TMB. So, moving ahead, I would like to hand over to Dr. Arun, who's our second speaker for tonight. Also, I would like to request to inform everyone that we'll be taking up question and answers at the end of the session. So you can just type it in the chat box which has been provided. Over to Dr. Arun.
Thank you. Thank you, Dr. Nirmal. That was excellent in one word. So it covered everything from clinical trials to the biology of every marker. So I'm sure it was very useful and exciting for all the participants. The TMB harmonization project, the paper got published very recently. It was we have a lot of learning to do from that too, in terms of validation of the TMB assay that we use. So with that, I'll just go on to my topic. And as Geeta just mentioned, you can drop in your questions in the chat box. My topic is going to be pretty short, where I'll be speaking about upcoming or next generation biomarkers that can predict the response to cancer immunotherapy. What Dr. Nirmal had talked is bodies and various FDA approvals and all are there, especially for PDL1, TMB, and MSI. Now, these are all markers that are still under evaluation that we have, researchers have identified while doing retrospective data analysis and incidental findings, etc. So, going back to the basics, just to set the stage, as Dr. Nirmal had initially shown, any interaction, the whole background of immune checkpoint inhibitor therapy is revolving around the interaction between a tumor cell and a T cell, where you have three major interactions: the B7 and the CTLA4 interaction, and the PDL1 and the PD1 interaction, which leads to inhibition of the T cell, whereas the HLA or the MHC molecule and the T cell receptor interaction, where the HLA presents the neoantigens to the T cells, lead to the T cell activation. So the immune therapy strategies revolve around blocking this inhibition signals. Particularly, you have antibodies against monoclonal antibodies against the PD1 or the PDL1 or the CTLA4. All these antibodies can bind on to the specific molecules and prevent the inhibition signal and activate the activation signal of the T cell. So with this background, there is this whole concept of tumor microenvironment, where as we study more and more deep into the tumor tissue per se and the pathways involved in it, we find that there is a skewness in the certain pathways that are upregulated or downregulated in people who respond well to the immune therapy. So to give you an idea, for example, consider this to be the T cell and this to be the tumor cell. This is the interaction that I just mentioned. There are factors within the T cell. For example, the T cell effector function is totally dependent on what is known as the stemness of the T cell, and that is in the sense that the T cell should have enough number of stemness where it can generate more number of T cells of it, and the downregulated T cells, that is the T cells that have got exhausted due to the activity, should be less. So this delicate balance between the stemness activity and the exhaustion activity in the T cell is pretty critical in in determining how well the T cell effect on the tumor cell is present. Likewise, there are also factors within the tumor cells, such as immunogenic tumor antigens, the type of neoantigens that are presented to the T cell, tumor mutation burden, mismatched deficiency, which we just saw, and the process of antigen processing and presentation, which involves complex immunological pathways that involve the JAK-STAT pathways, etc. And immune checkpoint upregulation, that is the PDL1 upregulation, is also pretty important for us to have a proper anti-PD1 therapy. So these are certain factors that improve the activity of the T cell on the tumor cell. Whereas there are also other pathways, especially which revolve around the tumor microenvironment. For example, in any tumor tissue, you're not just going to have the tumor cell, you're going to also have the immune cells, particularly the CD8 cells. Associated with that, you will also have certain cells such as regulatory T cells, fibroblasts, macrophages, myeloid-derived suppressor cells, etc. Now, particularly in the tumor evasion pathways, such as oncogenic pathways, which lead to STK level loss. We'll talk in detail about this in the coming slides. Copy number alterations, chromatin modifiers, all these lead to certain suppression of the T cell activity. Likewise, immunosuppressive effects of TGF beta signaling pathway is one of the very important reasons why certain people, even though they have good TMB, do not respond well to immune therapy. So the factors that can increase immunological response in the tumor tissue, the factors can be from the T cell or the tumor cell. Now, the T cell factors include intratumoral infiltration. How much the T cells are infiltrating the tumor site is a very critical factor, and that is totally dependent on the transcriptional signatures of the CD8 cells or the T cytotoxic cells. And enhanced effector function of the T cells are also required, where increased expression has been noted in such good cells where PRF1 granzymes and CD8A genes are all expressed. Increased clonality. So a clonality refers to how many different clones of T cells are there in the tumor tissue. So the clone is nothing but a T cell which is specific to one particular neoantigen, or to be more specific, one particular epitope of the neoantigen. As we would know, the T cell receptor is highly specific to the particular epitope. So in range of clonality, if the tumor tissue has a clonality of 1, it means that it has just monoclonal population. So monoclonal means it doesn't have a diverse response. It doesn't there are no diverse number of T cells present in the tumor tissue. So increasing the clonality is another factor, and greater stemness. So stemness and exhaustion of the T cell are two mutually independent, two interrelated events where they are exclusive to each other. So expression of CXCR5 and transcriptional factor TCF7 leads to stemness, where more number of T cells can be generated, whereas presence of TIM3 and CD39 leads to suppression of the T cell, and that leads to T cell exhaustion. So that means the number of T cells that are being produced drastically reduces. So this balance is also pretty important. And the tumor cell response mechanisms include the number of neoantigens that are produced. And in cases of viral mediated cancers, the viral antigens are also important. And increased tumor mutation burden, something that we have seen in the previous lecture very exhaustively. And immunogenic alteration. So inactivation of certain genes such as SERPINB3 or SERPINB4 leads to improved immunogenic response. Genomic gene itself is a good factor of the tumor cell that leads to better response to the immune checkpoint inhibition. And lastly, I'll talk about chromatin modifier loss. It's very important, and almost there are number of anecdotal evidences to show that certain chromatin modifiers like ARIDA1A or SMARC-A4 mutations in these genes actually lead to higher TMB and better response to the immune checkpoint inhibition. Now, there are also factors that reduces the immunological response at the site. So the tumor cell response include, tumor antigens. So the neoantigen in the tumor cell, if that mimics a lot to the self antigens or the self proteins, then the immune cell response to that becomes very, very limited, because the immune cell thinks that, okay, this seems to be very similar to the self protein. So as to prevent autoimmune response, the immune cell does not elicit a very, very strong response. And deficient antigen presentation is also another very key factor, which is coming up as a very good marker, particularly related to beta-2 microglobulin, HLA, JAK-STAT pathway, and to the interferon gamma pathway. I'll talk about this in the next slides. Oncogenic pathways, such as pathways including STK11 PTEN mutations or the EGFR mutations, beta-catenin gene pathways, and the mutations of genes in those pathways have a direct effect on the response to the tumor cell to the immune checkpoint inhibition. And immune evasion alterations and copy number alterations are known to cause, again, resistance mechanisms to immune checkpoint inhibition. End of the day, they all lead to pathway alterations where the T cells response to the tumor cell is very limited. There is also a tumor microenvironment where there are two major ones. One is the tumor suppressive stroma cells, and the other one is the fibroblasts or endothelial cells, and they have high level of TGF beta signaling, which leads to immune suppression. Likewise, higher number of myeloid-driven suppressor cells and regulatory Treg cells also lead to immune suppression at the tumor microenvironment. So, of all these factors, I will focus upon a few factors for which we have some good number of studies and evidences to show how they are responding to immune therapy. So, coming back to the basics, where here I will talk a bit about the immune pathway that is involved in the T cell and the tumor cell environment, tumor cell interaction. So, essentially, with the major histocompatibility complex 1, you have a protein called as beta-2 microglobulin, which binds with the HLA molecule, class 1 molecule. It could be HLA A, B, or C. And with that, the beta-2 microglobulin binds, and this beta-2 microglobulin and its diversity is a very critical factor that determines how well this interaction of the T cell receptor and the MHC takes place. So there have been studies which have shown that the loss of beta-2 microglobulin gene per se in the tumor cell or loss of heterozygosity of this beta-2 microglobulin has been demonstrated to show innate or acquired resistance to PD-1 and the CTLA-4 inhibitors. So especially it has been shown in anecdotal cases of melanoma or gastrointestinal cancers and also in lung cancers. Likewise, another important marker which is coming up is that the loss of HLA diversity. So as we go back to the basics of the HLA, each of us have 2 copies of the HLA, and if the HLA allele is different for in both the copies, then it shows that the person can elicit an immune response against a much more diverse set of antigens. Now, when there is a deletion of one of the HLA molecules, which leads to reduction in the diversity of the HLA molecule exhibited or expressed by the tumor cell, then that leads to immune evasion by the neoantigen. So essentially what happens here is that this MHC molecule gets deleted in the gene at the gene level. So the expression of the MHC itself is reduced or impaired, and the diversity of the MHC is also reduced. So that leads to corresponding poor activation of the T cell or poor presentation of the antigens to the T cell. And this expression of the HLA molecules is known to happen through the pathway of the interferon gamma. Now, the point reason I'm showing this interferon gamma in particular is that there are multiple studies which I'll show that show that the pathway of the interferon gamma and the genes in the interferon gamma pathway are differentially expressed in those who have resistance and who do not have resistance to the immune checkpoint inhibitor. For example, the T cell produces, what happens in this pathway is the T cell produces interferon gamma that binds to the interferon gamma receptor 1 and 2, which is there on the surface of the tumor cell. Once this binding happens, it activates the JAK, and that activates the transcriptional factor STAT. And the STAT transcriptional factor activates the production of a variety of proteins, including the IRF1, interferon response factor 1. And that leads to production of PDL1 and the the HLA molecule. So we might, if you remember the basics, we should know that we know that the expression of PDL1 as well as the MHC is very important for the proper interaction between the T cell and the tumor cell. So what happens here is that there are studies to show copy number alterations, specifically large deletions or copy number variants within the interferon pathway has been identified as a mechanism for checkpoint inhibitor resistance. So in one study in 2016, they found out of 12 melanoma tumors with primary resistance to anti-CTLA-4 therapy, 75% of them harbored genomic defects in the interferon gamma pathway. So some of the defects include copy number loss of the interferon gamma receptor or the IRF1 gene or the JAK2 gene, and amplification of interferon gamma pathway inhibitors such as SOX1 or the PASA4 or the PTPN2 genes. So these genes and their corresponding proteins actually inhibit this interferon gamma pathway. So when these genes get amplified, the proteins get amplified and the interferon gamma pathway is inhibited, leading to lesser production of the MHC and the lesser expression of the MHC and the PDL1. So the take-home message here is that this interferon gamma pathway is very differentially altered, especially in cases of melanomas where there is a primary resistance to the immune checkpoint inhibitors. So apart from this, you also have mutations in oncogenic signaling pathways that are known to be associated with the immune evasion of the tumor cell. For example, in lung adenocarcinomas, the very common tumor suppressor gene called STK11, it's also called LKB1, they have been shown to have reduced, wherever there is an inactivation of STK11 because of, deleterious mutations in the STK11 gene, it shows that it has been shown to have a reduced number of tumor infiltrating lymphocytes and hence a worse response to immune checkpoint inhibitor. In multiple studies have shown that. So now that it's almost majority of the oncologists, when they see a STK11, they do know that the patient might not respond to immune checkpoint inhibitor as much as a different patient. Likewise, the loss of PTEN also has a very similar activity in various tumors. Apart from this, drivers of clones, such as EGFR mutation or ALK mutations, they also tend to block the immune response. For example, both human and murine models of EGFR mutated lung tumors displayed very lower number of infiltrating CD8 cells compared to a KRAS mutated lung tumor. And a large meta-analysis, it showed that those people with EGFR mutated lung cancers did not respond as well with PD-L1 therapy, and their response was not as different as anybody who is undergoing chemotherapy. So bottom line is EGFR mutations are indeed a robust predictor of PD-L1 blockade resistance. Whereas it's not the case with KRAS. I'm not going to the details of it. KRAS is again a very huge story where, interestingly, they have found that same KRAS mutation in a lung cancer behaves differently than the KRAS mutation in a colorectal or a pancreatic cancer, and the tumor infiltrating lymphocytes behave differently. So in KRAS mutated tumors, you do have many other pathways or many other factors which are yet to be identified to see how the response to immune checkpoint inhibitors are. And lastly, the chromatin remodeling complex. It's a very important pathway where the histone particles are all affected because of these specific genes. So ARIDA1A is one such very important chromatin remodeling complex protein, and it was found that in mice with this ARIDA1A deficient ovarian tumors, PD-L1 inhibition led to reduced tumor burden and prolonged survival because whoever had the mice that had ARIDA1A deficiency responded much better to PD-L1 inhibition. So ARIDA1A loss and impaired ARIDA1A binding. So the hypothesis that the authors went on to say is that whenever there is a loss of ARIDA1A, it leads to binding of the MMR proteins such as MSH2, and they reduce the MMR activity, and that leads to higher mutational frequency and leading to higher amount of neoantigens being produced. So that again is a good thing for the cancer wherein immune therapy will work. So lastly, I will go on to a very interesting concept that is coming up, which is called as the cold and the hot tumors, where cold tumors are those where you don't have much of tumor infiltration with the immune cells. So for example, here, these violet ones are all the tumor cells, but you don't see much of T cells or the dendritic cell, very heavy infiltration of the immune cells to the tumor site. And that helps the, so essentially these hot tumors are the ones that can actually respond very well to tumor anti-PD-1 or any immune checkpoint inhibitor therapy. So there are strategies that are being adopted to identify how a cold tumor can be converted to or can be changed to a hot tumor. So they call it the strategies of heating up of the tumor microenvironment. So of the 4 strategies that are commonly studied upon, these are all still under animal model studies, where you can see that one of the models is to boost the cross-presentation of dendritic cell with CD8 cells. So essentially, dendritic cells help the CD8 cells to mature, and when you prime the dendritic cells, they in turn prime the CD8 cells, and the CD8 cells, once they are activated, they can help. They can work in killing the tumor cells. The second strategy is metabolic reprogramming, where a tumor cell or a T cell, a T cell or a macrophage which has not metabolically active, there are mechanisms which are genetic, CRISPR or related gene alteration mediated mechanisms, where they increase the metabolic activity of the cell, and whereby the cell has more number of mitochondria inside it, and they get metabolically active, and they are able to protect against the tumor in a much better fashion. So there are certain molecules that have been identified that can do this metabolic reprogramming. Likewise, the third mechanism is targeting the epigenetic modulators, where it can either promote immunogenicity of the tumor cells, or they can re-educate the tumor as the T regulatory cells or the tumor activated tumor associated macrophages, and they can support the in such a way that you either block or overexpress certain genes in such a way that helps in the fight against the tumor cells by that CD8 cells. Lastly, there are also mechanisms where virus-mediated antigen presentation takes place, where the virus is genetically engineered, and the virus can have certain key molecules of the neoantigens of the tumor cells, and they in turn activate the CD8 cells, such that the CD8 cells are much more activated and they go on to kill the tumor cells. So these are all these are all studies that are still under in the laboratory stage, and some of them are coming up into a probable strategy where you can convert a cold tumor into a hot tumor. So lastly, to sum up, these are the consolidated list of biomarkers. So well-established markers include TMB, PDL1, MMR, and MSI. So these are all well established, and you do, as I mentioned just now, you do have HLA class 1 diversity. More the diversity is associated with a positive response to the tumor, immune checkpoint inhibitor, and copy number variants, tumor T cell inflamed microenvironment, TGF beta expression, mutations in the beta catenin pathway, STK11, POL-E, or the ARIDA mutations. All these are all markers that are still being investigated, and I'm sure in the next few years we should be able to see well-established markers, and we are going to have subset of patients who might be whom we might still be able to classify in a much better fashion than the way we are classifying them as responders or non-responders to immune checkpoint inhibitors. So the future, this is called as a basket trial, wherein you can have a case of breast cancer or a colorectal cancer or a prostate cancer. You look for the specific tumor markers such as TGF beta or expression of interferon gamma. And once you have that marker, maybe based on that marker, the extra drug can be predicted. For example, if there is a high TGF beta expression, you can have a TGF beta inhibitor along with PDL1. Or if you have a low interferon gamma, you can give a STING molecule, which is essentially stimulators of interferon gamma, and STING agonist can be given along with a PD1 inhibitor. So the future may be this, where along with immune checkpoint inhibitor, you do identify certain immune markers, and those markers and the corresponding drug molecules will be given in combination to have a much better response to immune checkpoint inhibition. These are my references, and I thank you all for this opportunity. I'll be very happy to answer any questions.
Thank you, Dr. Arun. That was an amazing talk. So we'd like to welcome question answers from the audiences. You can type your questions in the chat box and we'll be answering it.
As per your, Dr. Nirmal, it's a question to you.
Yeah.
As per your clinical experience, which biomarker you feel is more apt to do on or any priority or preference in combining TMB, PDL1, MSI together for immune therapy drugs?
Okay, so based on my experience, which biomarker you feel is more apt, or do you priority or prefer in combination? Yeah, so usually you go by PDL1 and MSI, and TMB is still not freely available. So, but TMB also I kind of do for all my patients. So PDL1, MSI, yes, and TMB plus or minus, that way.
So I do have a question for you, doctor.
Yeah.
You would have come across mutationals, such as ARIDA1 mutation or the STK11 mutation. I'm sure you have come across that in your patients. So have you had any experience, or how do you judge for an immune therapy based on those mutations? How as an oncologist you see those mutations for immune therapy, doctor?
ARID1A, the data is a little, not very clear. There is a paper by Razel Kruzrok where she says that ARID1A mutations, they express TMB and MSI, and based on that, she says that these tumors respond very well to immune checkpoint inhibitors. But there are some cases where ARID1A is so not all ARID1A mutations are the same. So I think it's based on some particular mutations which make it more immune responsive, and some which don't. Yeah.
Yeah. Okay. Now, as it goes on, I think we are going to subset the patients more and more.
Yeah. Yes. Yeah. Right. Like STK11, KEAP1, these are also important mutations that we have to look for in lung cancer.
There are no further questions.
Yeah.
We can wrap up if there are no further questions.
Yes, so we can wrap up. So, Dr. Nirmal, Dr. Arun, on behalf of MedGenome Labs, I would like to thank you for being our speakers today. And I would like to thank everyone for attending the webinar. And, thanks to the organizer, everyone. And yeah, so thanks a lot, and we would wrap up our webinar, and the video is available on our YouTube channel, and we'll be sharing the link with the people who have registered. So thanks a lot again.
Okay, thank you.
Thank
Thank
you.
you.